Everyone. My name is Kirti Bilgi and my presentation topic is biofilms. So what are biofilms? Next. So biofilms were first observation of surface associated bacteria was done by the Antony van Leeuwenhoek, as we all know about it in 1684. But the term biofilm was coined by Costerton in 1978, and in 1999 it was defined as a structured community of bacterial cells which are enclosed in a self-produced polymeric matrix which are adherent to the surface. So biofilms actually is a you can say a colony of multiple microorganisms, whether it is from same species or a different species, which are adherent to any non-motile surface. So bacteria exist in two different forms, as we know. That is one of the planktonic state, which is a free floating state, and the sessile state, which is adherent to the surface, both of which have existed on earth since first bacteria evolved. So bacteria are able to grow adhered to almost every surfaces, biotic or abiotic also. So in biotic we can say as a human body, and inside our human body also the microorganism grow on skin, they grow inside in any organ, if it is undergone on any replacement, like a pacemaker in heart, so there also they grow. So it kind of a biotic growth. And abiotic growth, it can grow on catheter also in hospitalization, and in industrial area, in plants also they grow, so it is kind of a abiotic adherence. So they are forming architecturally complex communities termed as a biofilms. So in biofilms, cells grow in multicellular aggregates and are encased in an extracellular matrix produced by the bacterial themselves. So attachment of bacteria to the surface result in rapid alteration in the expression of number of genes responsible for exopolysaccharide, that is called as EPS, or slime production and maturation. So as soon as the bacteria attaches to the surface, there is a change in the physiology of the bacteria. The group of bacteria will start showing the changes in their physiology and in the changes in their gene expression, so as to which favors the growth of the colony. So this results in the production of a protective barrier that protects the bacteria against the organism's endogenous defense system or an external agent such as the antibiotics. Next. The structural component of biofilms, the structural how it looks, the biofilms, the molecular mechanism that regulate biofilm formation is vary greatly among the different species, and even vary between different strains of the same species. However, some features are recognized as general attribute of biofilm formation. For instance, all biofilms contain an extracellular matrix that holds cells together. So it's a slime-like product that bacteria secrete, which can hold them together. This matrix is often composed of a polysaccharide biopolymer along with other components such as the proteins or the extracellular DNA. The nature of the matrix exopolysaccharide greatly varies depending on the growth condition, medium, and the substrate. So, Ko et al. In 2007 indicated that biofilm can be formed by microorganisms to cope with local environment challenges. Things like pitting in piping or a nick can provide the desirable environments. The microbial cells may seek out small chambers or cavities, assemble there, and engage in quorum sensing behavior. So as we know that biofilm formation, it is like I said before, the substrate or the abiotic nature or the biotic substrate there where they form a biofilm, like in the tooth cavities, in dental caries and cavities also they form a biofilm. So they seek out the small chambers and cavities where they can establish themselves without getting affected by the external environment. So they search for that a safe place for development. Then the main component of this, or the you can say the main behavior of this biofilm is based on the quorum sensing, which is defined as a regulatory mechanism of microorganisms that involves the release of molecules which, when present at threshold concentration, signal the expression of bacterial genes controlling specific group of actions such as formation of biofilms. So there are many different reasons that microbes may seek out these chambers or cavities. Some of the reasons include the environmental condition, chemical stress, variable temperature, fluid flow, and limited supply of nutrients. Next. Next, so as you can see in the picture, the where the biofilm develops. So one of the first picture you can see the biofilm formation in water pipeline. So it is a common phenomenon, formation of biofilm in a water pipeline. So you can see in the regular water supply pipelines there is a growth of the algae. So it's a kind of a biofilm formation only. And second, you can see as a biofilm formation in a medical devices, which is nowadays which is a common threat in the medical industry. So you can see the instrumentation device. It can contact lens. It can form a biofilm on contact lens. Then urinary catheter, which is an example of E. coli biofilm formation in the urinary catheter, prosthetic joints, then coronary stents like a pacemaker and intrauterine devices also they can form the biofilms. Next. So what is the quorum sensing? So in biology, quorum sensing or quorum signaling is the ability to detect and respond to cell population density by gene regulation. So quorum sensing is a type of cellular signaling and more specifically can be considered a type of paracrine signaling. However, it is also contains the traits of both autocrine signaling. A cell produces both the autoinducer molecule and the receptor for the autoinducer. As one example, the quorum sensing enables bacteria to restrict the expression of specific genes to the high cell densities at which the resulting phenotype will be most beneficial, especially for phenotypes that would be ineffective at low cell densities and therefore too energetically costly to express. Second, the many species of bacteria are use quorum sensing to coordinate gene expression according to the density of their local population. So quorum sensing in pathogenic bacteria activates host immune signaling and prolongs host survival by limiting the bacterial intake of nutrients such as tryptophan, which further converted in serotonin. So like in quorum sensing they will secrete some molecules, so which will help to promote their growth. Like if it is in pathogenic bacteria, the quorum sensing will activate the host immune signaling and prolongs the host survival. So the bacterial biofilm will survive as long as the host survives. So what they do, they secrete such molecules which will activate the host immune system, which will attack the biofilm, and there will be so that the host is survived. The biofilm will not take over the host. The host survives, and as long as the host survives, the biofilm also survives. The third, quorum sensing allows the commensal interaction between host and pathogenic bacteria. So quorum sensing may also be useful for cancer cell communication. Next. So quorum sensing discovery. So quorum sensing was first reported in 1970 by the Kenneth Nielsen, Terry Pat, and J. Woodland Hastings, who observed what they described as a conditioning of the medium in which they had grown the bioluminescent marine bacterium Alivibrio fischeri. These bacteria did not synthesize luciferase and therefore did not luminescence in freshly inoculated culture, but only after the bacterial population has increased significantly because they attributed this conditioning of the medium to the growing population of cell itself. They referred to the phenomenon as auto-induction. Some of the best known example for quorum sensing come from studies of bacteria. So bacteria use quorum sensing to regulate certain phenotype expression, which in turn coordinate their behavior. Some common phenotype include the biofilm formation, virulence factor expression, and motility. Certain bacteria are able to use the quorum sensing to regulate the bioluminescence, nitrogen fixation, and sporulation. The quorum sensing function is based on the local density of the bacterial population in the immediate environment. It can occur within a single bacterial species as well as between diverse species. So the funda of this quorum sensing is totally is based on the cell density. At lower cell density, the cell or the cells involved in the biofilm will not secrete some molecules, but as soon as the cell density increases, it will start showing the that different phenotype, which will help to grow. Next. So as you can see the mechanism of quorum sensing in gram-negative bacteria. At low density, you can see there is less cell and there is less product also. As soon as the cell growth increases, the cell density increases, you can see the molecules also, the acyl homoserine lactose autoinducer. The autoinducer also increases. So you can see in this how the cell density increases depending upon the quorum sensing mechanism. Next. The role of quorum sensing in biofilm development. When aggregated in high enough densities, some bacteria may form these biofilms to protect themselves from biotic or abiotic threats. The biofilms may also serve to transport nutrients into the microbial community or transport toxins out by means of channels that permit the extracellular polymeric matrix like cellulose that holds the cell together. Finally, the biofilms are an ideal environment for horizontal gene transfer through either conjugation or the environmental DNA that exists in the biofilm matrix. The process of biofilm development is often triggered by environmental signals. The bacteria are proven to require flagella to successfully approach a surface, adhere to it, and form a biofilm. As cell either replicate or aggregate in a location, the concentration of the autoinducers outside of the cell increases until a critical mass threshold is reached. At this point, it is energetically unfavorable for intracellular autoinducers to leave the cells, and they bind to the receptors and trigger a signaling cascade to initiate the gene expression and begin secreting an extracellular polysaccharide to encase themselves inside. So when the new cells approaches to the pre-developed biofilm, so how they are engulfed inside the biofilm, how they become a part of the existing biofilm. So as soon as they reach the biofilm, which is saturated with the already autoinducers, they start they cannot secrete the autoinducer outside of the cells. So what happens, the cells have the receptors for those autoinducers on themselves, so that the autoinducer will bind to the receptor and signal a pathway that it doesn't need to secrete the autoinducer outside the cell. So it will signal a pathway and start some another mechanism by which they secrete the extracellular polysaccharide. That is it can be a cellulose or it can be other polysaccharide. So they secrete that extracellular matrix, and hence they can attach themselves to the existing biocell which is having the same extracellular polysaccharide. Okay, next. So here you can see the formation of biofilm in a stepwise manner. So it is in the first stage is the planktonic bacteria, which is a free swimming bacteria that can reversibly attach to a different type of surfaces. The second part is the auto-aggregation. Bacteria transition from reversible to irreversible attachment due to the EPS production. EPS, that is the extracellular polysaccharide production. When bacteria start first are free flowing or about to attach a surface, first they will attach to each other. That is, the bacteria, the free flowing bacteria directly doesn't don't attach to the surface. They first attach to themselves, to each other. They form a aggregate, and then they will attach to the surface. As soon as they attach to the surface, they will start secreting this EPS, which forms which forms the protective environment. Then the establishment of the bacteria. Early development of biofilm architecture into the stable supercellular structure. The cell density start increasing step by step manner. Then the growth happens. The late development of microcolonies, multiple species, secondary colonization by multiple species can occur. So here you can see if the bacteria biofilm has started with the one species, it is no need to say that it will consist only one type of a species or one type of a strain. The bacteria can be a different type of a strains and the different types of a species also. So the mature biofilm, once the growth happens, the biofilm matures, and you can see when the antibiotic attacks the and it is attacked by an antibiotic, there is a gradation of antibiotic. The antibiotic will go inside the biofilm, or it cannot enter the biofilm. There are two types of possibilities also, and the microorganism present in the biofilm react differently to the antibiotic. Maybe they will get resistant to the antibiotic also. The mature biofilms are characterized by complex 3D structure antibiotic gradients, and the antibiotic gradients are indicated different size blue dots. In this you can see in these images. Next, the The extracellular matrix of biofilms also harbors adhesive proteins. For instance, the SORES matrix harbors the biofilm associated proteins termed as BAP that are required for biofilm formation. These proteins are found anchored to the cell wall of the SORES and serve to hold cells together within the biofilm, probably by interacting with other proteins on the surface of neighboring cells. In certain strains, the expression of BAP proteins eliminates the requirement of extracellular extra polysaccharide for biofilm formation. So, as I said earlier, so there are some proteins present on the bacterial cell wall itself, which will attach to each other. So every time there is no need that the cell should secrete the extra polysaccharide, but they can attach to the surface by attaching themselves also. So in case of Bacillus subtilis, which is gram-positive bacteria, the expresses a single major protein associated with the extracellular matrix termed as a TSA. The mutants which are deficient in this TAS-A fail to form biofilms, despite the fact that they can still produce exopolysaccharides. The TAS-A has recently been shown to form the extracellular filaments that have amyloid-like structure properties and is thought to play a small architectural role in the extracellular matrix. The TAS-A is not the first amyloid-like protein to be implicated in biofilm formation. So, as you can see in SORES, in Bacillus subtilis, there are two different types of a mechanism for the adherence. So now in E. coli, the curly protein also forms the amyloid filaments and is critical for biofilm formation. Other proteinaceous structures important for biofilm formation are pili and fimbriae. These cell appendages are used to adhere cells to each other or to different surfaces. E. coli produces type 1 fimbriae that are required for the adherence to mannose containing receptors. These fimbriae are important for biofilm formation on plastic surfaces as well as in the host cell during the urinary tract infections. Next.
Wait, wait, wait a minute.
Next.
Wait a minute.
Next. हाँ सर.
Okay, continue.
Okay, continue. हाँ सर. Sorry. So as we had seen in E. coli, now in the Pseudomonas aeruginosa also has many surface proteins that contribute to biofilm formation. For example, mutants unable to produce type 4 pili or the cup A fibriae were defective in surface adhered biofilms. In addition, there are other matrix associated lectin binding proteins that recognize and bind carbohydrate moieties. This facilitates the cell matrix or cell to cell interactions within the biofilm. Pseudomonas aeruginosa has two lectin binding proteins involved in biofilm formation, the LEC A and LEC B protein. In addition to the exopolysaccharide and proteins, extracellular DNA also provides structural integrity to the biofilms. The biofilm matrix of Pseudomonas aeruginosa contains significant amount of eDNA, which is extracellular DNA. In addition to DNAs, to culture inhibits biofilm formation and dissolves mature biofilms. S-Aureus biofilms also have eDNA in the matrix and it functions to provide stability to the biofilm. It is thought that this eDNA is caused by cell lysis and subsequent release of genomic DNA. So what happens within the biofilm? The cell dies, it releases the cell content, it releases its DNA, and as such, by this mechanism, you can find the DNA also in the biofilm. Next. Detachment of cell clusters. Now, after the formation and the maturation of this biofilm, the detachment also happens. The detachment of biofilm cells are a concern in pharmaceutical manufacturing as well as in clinical settings. However, it is one of the least studied areas of biofilm processes. New methods using digital time-lapse microscopy along with flow cells designed for biofilm provides opportunities to study this mechanism of detachment. The detachment is comprised of two processes, erosion and slugging off, based upon the magnitude and frequency of the detachment event. The Studley et al. 2001 describes erosion as a continual detachment of single cells and small portions of the biofilms without specifying size distribution and frequency of biofilm particles. The author describes slugging as a rapid and massive loss of biofilm. So using digital time-lapse microscopy he was able to track single cells that were moving between the biofilm cells and observe the behavior and movement of the mature biofilm over the course of several days. over the solid surface in situ. The studies conducted showed a steady detachment of cell clusters over the monitoring period. The quantity produced up to 3.6 into 10 raised to 9 CFU per minute. Next. Now, biofilm, the role of biofilms in different areas. So, next. Biofilms in medicines. It is suggested that around two-thirds of bacterial infections in humans involve the biofilms. The infections associated with the biofilm growth usually are challenging to eradicate. This is mostly due to the fact that mature biofilm displays antimicrobial tolerance and immune response evasions. The biofilms often form on the inert surface of the implanted devices such as catheters, prosthetic cardiac valves, and intrauterine devices. Some of the most difficult infections to treat are those associated with the use of medical devices. The microbial infections can develop on all medical devices and tissue engineering constructs. 60 to 70% of hospital-acquired infections are associated with the implantation of the biomedical devices. The level of antibiotic resistance in the biofilm is much greater than that of the non-biofilm bacteria and can be as much as 5,000 times greater. The extracellular matrix of biofilm is considered one of the leading factors that can reduce the penetration of antibiotic into the biofilm structure and contribute to the antibiotic resistance. Further, it has been demonstrated that the evolution of resistance to antibiotics may be affected by the biofilm lifestyle. It has been shown that the introduction of a small current of electricity to the liquid surrounding a biofilm together with a small amount of antibiotic can reduce the level of antibiotic resistance to the level of non-biofilm bacteria. This is termed as bioelectric effect. The application of a small DC current on its own can cause the biofilm to detach from its surface. A study showed that the type of current used made no difference to the bioelectric effect. Next. So biofilms in industry. So biofilms can also be harnessed for constructive purposes. Example, many sewage treatment plants include a secondary treatment stage in which wastewater passes biofilm grown on filters, which extract the digest organic compounds. In such biofilms, bacteria are mainly responsible for removal of organic matter, while protozoa and rotifers are mainly responsible for removal of the suspended solids, including pathogens and other microorganisms. Slows and filters rely on biofilm development in the same way to filter surface water from lake, spring or sources for drinking purposes. What can be regarded as water is effectively a waste material to these microbial organisms. Biofilms can help eliminate petroleum oil from contaminated oceans or marine systems. Oil is eliminated by the hydrocarbon degrading activity. This species of hydrocarbon is oil Biofilms are used in microbial fuels to generate electricity from a variety of organic waste and renewable bio. The biofilms are also related to the improvement of metal resolution in bleaching industry. The aggregation of micropollutants for UV environment. Next. Biofilm food industry. Role of biofilm in food industry. So biofilm are problematic in several food industries due to their ability to reduce animal protein. Bacteria can survive long periods of time without animal matter and oil because the biofilm remains on the plants or in the processing equipment. The growth by biofilms can affect the heat flow across the surface and increase friction resistance. This can lead to increased energy consumption. Heat from the surface of the produce inner can lead to the skin tissues, foodborne infections such as tuberculosis. The latest proposed biomolecules presenting marked antibiofilm activity include range of metabolites such as bacterial, lipids, even plant and animal alcohol. So already glycol where the advertiser has no the materials which are biofilms. Biofilms are used in in shellfish and algal aquaculture. Biofouling, microbial species tend to block nets and cages and ultimately outcompete the farm species for space and food. The bacterial biofilms start the colonization process by creating the microenvironment that are more favorable for biofouling species. In the marine environment, biofilms could reduce the hydrodynamic efficiency of ships and propellers, which lead to the pipeline blockages and sensor malfunction, and increase the weight of appliances deployed in seawater. The numerous studies have shown that biofilm can be a reservoir for potentially pathogenic bacteria in freshwater aquaculture. Moreover, the biofilms are important in establishing infections on the fish. As mentioned previously, biofilms can be difficult to eliminate even when antibiotics or chemicals are used in high doses. The role that biofilm plays as a reservoir of bacterial fish pathogens has not been explored in detail, but it certainly deserves to be studied. Next. So these are the some of the methods, existing techniques to study the biofilm growth. So there are different types of method which employs the different kinds of devices to study the biofilm growth. So some methods are the like flow cell, microfluidics, optical sensors, or electrochemical sensor or mechanical system, which target the microbial biofilm growth, antibiotic resistance, and eradication procedures. So there are also you can say the pros and cons of this method which are listed in the table. Next. Now, there are the sensors to investigate the biofilm dynamics. The biofilm dynamics, that is the physiology of the biofilm. So, there are sensors too. We can, for the research purpose or for the study purpose also, they have developed the sensors which can investigate or the track the biofilm dynamics. So, the principle behind that are the electrochemical sensors, the optical sensor, mechanical system. Like with the example for the electrochemical sensors are microelectrode probes which target the oxygen, the difference in oxygen or the production in oxygen, depletion in oxygen, the differences in pH, ions, temperature, and metabolites, which is sensitive and easy to use, but it is invasive. Analysis can easily comprise the structure of the biofilm with the potential cross sensitivity. The optical system, the example of the optical system is the planar optodes labeled micro nanoparticles, which again targets the oxygen, pH, heavy metal production, and metabolite production of by the biofilms. The mechanical system includes the interfacial rheometry and the tensiometry, which targets the interfacial material parameters for adhesion properties. So these are different types of methods which they have, which the scientists are applying for the investigation of biofilm dynamics. Next. The characteristic of the biofilm based sensors. So what kind of sensors they are using? As I said before also, it was like electrochemical sensors they use, the optical sensors, and micro gravimetric sensors. So there are different kinds of sensors which are based on the fluorescence, bioluminescence. So these are the techniques that they use to study the biofilm. So as I said earlier, some of the microorganisms previously they don't show the bioluminescence, but as soon as the biofilm grows, the mass grows, the cell density grows, they start showing the bioluminescence. So these all the optical devices track the track this bioluminescence, development of bioluminescence, to study the growth and development of the biofilms. So that's it, all my friends and sir. So thank you so much for this. Thank you.
Thank you.